Hello, everybody. We're the University of New Mexico Team 1, and we'll be presenting a pathway towards global scaling for BennuBio. My name is Jesus Ortega, along with my teammates Nikhil Reddy, Bhuva Ramalingam, and Irma Vazquez, and our advisor, Professor Peter Boroviev. We will now start with the company and product profile. BennuBio operates in the pharma and life sciences technology sector under the drug discovery and R&D industry. It is a privately held company looking to scale globally and not actively searching for acquisitions yet. For flow cytometry applications, the VeloCyte represents a disruptive technology which applies acoustic diagnostics to perform cell analysis. This novel technology allows for samples to be analyzed up to 100 times faster than the current standard, while keeping the samples intact for reusability. In terms of strengths of the company, the BennuBio team has a strong technical skill set in the area, and it has a strong link between the University of New Mexico and Los Alamos National Lab. Similarly, the technology developed has a potential to be disruptive in the market, as the VeloCyte has an estimated TRL of 8. On the other hand, for the company's weaknesses, the company is based around their only product, the VeloCyte. And so far, they have been able to raise only $7 million, from which 5 came directly from a venture capital group. Moreover, our team only identified very few publications highlighting the applications of this technology. Lastly, it should be mentioned that the VeloCyte has an MRL estimated of 4, which means there's not a production line in place at the moment. Moving on to the opportunities. According to the Boston Analytics report provided, a decrease in new companies entering the sector could mean that the barriers to entry are placed too high, or that older companies can be absorbing the new companies. However, this can translate into less competitors for us. According to a market report, the compounded annual growth of the flow cytometry market is expected to grow 8.3% annually, while highlighting that the Asian Pacific and North American markets are the ones growing the fastest. Furthermore, according to the same report, Asia Pacific only holds 5% of the companies in the sector, while Europe holds about 15%. In the same report, the funding momentum for the sector appears low. Nonetheless, it appears high for the flow cytometry market. Lastly, we should recall that the National Institutes of Health funded BennuBio with $2 million, and this can be used as leverage to hopefully be able to continue getting R&D funds. Moving into the threat section, there are industry-wide standards in place for flow cytometry techniques, which can be an entry barrier for the VeloCyte. Similarly, since BennuBio is located in the US, according to the report, over 70% of the companies in the sector are located within the same region. The industry in which BennuBio operates has the largest number of companies according to the Boston Analytics report. Moreover, there was a drastic reduction in the funding rounds for the sector in 2020. However, the team attributes this to be due to the global pandemic in hopes that more funding will be available for 2021 and on. Lastly, while the number of acquisitions can be keeping new entrants out of the sector, it should be noted that Thermo Fisher Scientific and Agilent Technologies are both direct competitors to BennuBio, but also major acquirers in this sector. Now, applying what the team has learned from the SWOT analysis and the market analysis, a 3-step scaling strategy has been developed.
To begin, we want to show you a simple schematic of our global scaling roadmap where we have delineated 3 main steps with more detailed subtasks to reach our goal. In order for BennuBio to increase their assets and capabilities, the company should seek out funding from investors. We should recall that BennuBio has already received $5 million from Cowen Ventures, a firm in the USA and China investing in high-tech development for healthcare. Nonetheless, we strongly suggest to continue seeking out investments from firms that share BennuBio's vision. For example, Noble Holdings is a life science venture capital investing firm that invests in biotechnology and medical technology and focuses on innovative drugs, medical devices, and diagnostics. In 2020, they executed 79 investments with a portfolio of $2.3 billion. Similarly, Welcome is an organization with focuses on empowering ideas to improve health while helping researchers develop their careers with internships, scholarships, fellowships, and grants for up to $1 million for project funding. As engineers, we understand the burden of research and the time that is required to ensure that a project meets its milestones. For this reason, we recommend BennuBio to hire or appoint staff dedicated to finding and applying to potential funding sources. Since BennuBio has strong ties to UNM and Los Alamos National Lab, they should apply for grants that fund health science projects targeting NIH, NSF, UKRI, and many more government-funded and private institutions. Typical funding timelines for awards in 2019 and 2020 vary between 6 to 18 months. We should remember that BennuBio received $2 million from NIH to further develop the capabilities for the Velocyte. This could be used as leverage to apply for further R&D funding. It is paramount to demonstrate the capabilities of the Velocyte. In order to achieve this, BennuBio should actively seek out funding to continue their research efforts. R&D funding is necessary to continue evaluating the multiple applications of the instrument and disseminate as many results as possible. Publishing papers showcasing the capabilities and quality of results obtained using the Velocyte will help attract potential customers and investors, as they will be able to further understand the potential of the instrument to disrupt the flow cytometry market.
So analogous to acquiring funding, partnerships are equally valuable to ensure that Velocyte is validated and begins to adopted by important research institutions around the world.
Bhuva,
Also,
I'm having trouble understanding. Your audio is cutting out a little bit. Is that the same for everybody else?
Yes, it seems to be a problem. I can take over.
What you're saying.
Yeah, your audio.
Maybe Jesus can.
Yeah,
Okay,
that's
and
fine.
I paused your time, so this won't count
Okay,
against you.
no problem.
Just want to make sure the judges can understand everybody.
Yes, Bhuva, I'll take over for that, no problem. Analogous to acquiring funding, partnerships are equally valuable to ensure that the Velocyte is validated and begins to be adopted by important research institutions around the world. A common framework will act as a reference for partnership, not only for researcher-to-researcher interaction, but also for communication to persons in the related fields and professions. It will also facilitate the distribution of information and collaboration between the company and its customers. Some examples of renowned researchers who BennuBio could collaborate with are Dr. Hakeem Debala and Dr. Jennifer Gillette, both from UNM. Analogous to acquiring funding, partnerships are equally valuable to ensure that the Velocyte is validated and begins to be adopted by important research institutions around the world. A common framework will act as a reference for partnership. Whoops. Throughout this process, it is also important to identify influencers and opinion leaders which can help the Velocyte to become the next industry standard on flow cytometry. Two great examples are Dr. Bifina Litwin, who's a leader in analytical method validation and standardization of flow cytometry, and James Rothman, a Nobel laureate and chairman of the Department of Cell Biology at Yale. On the other hand, through collaboration, diversification will naturally occur, as there are many limitations that make traditional flow cytometers poorly suited for many applications, mostly due to the dilution that occurs during sample test. Clinically, flow cytometry is well suited that it can be useful to expand the operations in health research and treatment, which include monitoring the samples from COVID patients and those who have been vaccinated with it. By taking advantage of the Velocyte's unique capabilities, the instrument can be a candidate for laboratories worldwide, as they will be able to complete this analysis with much more shorter turnaround.
So as normal methods of advertising won't work for our product, we need to be creative and also selective while targeting customers. Our BennuBio team is relatively small with limited resources. So we need to find customers who will in turn will act as our potential sales representatives. Targeting leading research universities such as Carnegie's R1 research institutes and leading research labs will help us to take our device to the experts' hands in the field. And this will increase the validity of our product and also gives confidence and encouragement to others who work with them to try our product. Presenting at leading expos and also trade shows will significantly benefit us. One interesting option available for BennuBio is that to give license to the existing manufacturers like BioRad and BD Biosciences for a limited time basis to produce our product and sell them on a shared profit model. This model will help BennuBio to make profits on its own without having a production unit. It also helps us to take advantage of the company's existing communication channels and also customer base as well. So our next priority should be to reach out to institutions where the need for our velocyte is relatively less and are the areas that are difficult to reach us for now. This could include Carnegie's R2 research institutions and also universities in Asia, Africa, etc. Commercial labs and clinics like CVS Pharmacy and Tricor are also potential customers, but reaching out to them as first priority is risky given the price of the product and also everything and the need with them. With that, we thank you and we stand for any questions.
Thank you, guys. That was a great presentation. We are ready for questions. First one is, can you recommend a capture strategy for your early stage funding?
Excuse me, can you repeat the question once again, please?
Yeah, can you hear me?
Yes, we can.
Okay, can you recommend a capture strategy for your early stage funding?
Yes, so I think in terms of early stage fundings, like we said, it's important for the company to establish partnerships so they can invest or focus more of their attention to our R&D. So we think that instead of seeking out directly investments from venture capitalists or other companies, we feel like the velocyte needs to be further improved and, more importantly, validated in the field. Because if you want this instrument to disrupt the industry, it needs to be heavily validated so other research institutions like universities or national labs or so forth can understand the importance of having this type of instrument and what capabilities it provides, and similarly, what potential it has to expand to different applications. So I think it's imperative to focus their initial efforts to finding R&D grants from NSF or NIH, like we mentioned, so they can further develop and validate their instrument.
And just to add a little bit more, I think as of now we have technology that is worthy to invest more in, and also that has the potential to explore the market. So I think for now our team or BennuBio team should focus more on publicizing what we have already. So I think maybe attending more trade shows as expos or publicizing more scientific material on the advantage of our product will also help us to gain potential customers. We don't know who else is looking for a product like this and who are willing to invest in for a profit, like shared profit. So I think we need to advertise in the scientific world about the benefit of our invention. Thank you.
As Jesus told, we need a strong research with the universities, and this means to get more funding. At the same time, research in the sense they should specifically concentrate on the how they can improve the results they are getting from the device. BennuBio device, like machine learning kind of optimization, and basically with how they can market from the data. So they need to explore all these things. This will help to explore more collaboration and fundings. Yeah.
Thank you. What is the strategy for getting these researchers to try the new technology? What's in it for them besides the potential for faster results?
Well, it's honestly, that's their main sell point, because not only looking at the market right now, they can basically process fractions of a milliliter per minute, whereas the velocyte is able to process tens to hundreds of milliliters per minute. So it's a much faster application. Now, what is in it for them is the capability of being able to expand their research into different fields. For example, there are several limitations from flow cytometers in the market right now, mostly because they have to dilute their samples so much. And looking or opening applications such as cancer research and being able to do test trials and for vaccines, for example, trying to seeing the effects of a vaccine on a cell. These are things that can be done much faster, and we can open up fields that haven't been explored before.
I would like to add to that the Velocite is a great research instrument, and the way research is disseminated is mainly through research papers. So in targeting big name brand universities, Stanford, Harvard, and having research published in large scientific journals will prompt more scientists to also see the benefits of the Velocite, which not only includes the faster scanning speeds, but also the undamaged tissue cells, and undiluted samples.
So I just want to add up. So right now the researchers have been working on how basically they can use that flow cytometry to create some classification, machine learning classification, like differentiating whether the tested results are good or bad. So this is one kind of research which we have explored right now. So yeah, even the competitors like Thermo Fisher, they are actually working on that. But one thing which I can suggest is like we can do some predictions, okay, based on because these cells have a multiple parameters and a multi-dimension parameters, okay. So if we have that much capacity, we can predict more things. Like example, if based on that COVID blood cells, patient blood cells, we can discretize at what stage of time they are affecting and they are getting severized. We can also predict that. Yeah, that's it.
Thank you. What is the end game with these?
Can they be a little more specific? I see they have another follow-up question for that.
So Mr. Lavender said, what is the end game with these plans, and are there competitors?
Well, if they can be a little more specific with the end game, like what is the goal? If that's what you mean, well, the goal here is to be able to help the company essentially with the plan, so they can actually attract what we think is would be their priority customers, which are universities and other laboratories, essentially any type of center that does investigation. And that's the way that we think we should market it, as opposed to actually trying to sell through a website, like if we licensed it through Thermo Fisher or so forth. And to answer the other question, yes, we do have competitors. There are plenty. However, only Thermo Fisher Scientific has a device that utilizes the same acoustic wave technology. However, they have not been able to match the same amount of flow rate that BennuBio's device is able to process. So in that sense, at least in terms of numbers and how they can process the samples, they don't have a real competitor there. However, Thermo Fisher Scientific is definitely a much larger corporation that is probably willing to invest to compete against it or perhaps acquire.
Thank you. How will they support this beta testing and validation with limited manufacturing capabilities?
Well, that's basically why we are suggesting this beta testing, because BennuBio is not at the point where they have an MRL estimated of around 4. They're not at a point where they have a production line that can be sustained. So what we're suggesting is start slow, start partnering up with other potential customers, which could be universities or labs, and let them try your equipment. And if they're interested, then you can perhaps start establishing contracts with them so they can start acquiring the devices, so you can actually help you ramp up your production of these devices slowly. Then once you get it established, you can actually start targeting, like Nikhil presented in the second round, start targeting smaller clinics and other labs that could potentially benefit from having one of these devices on site.
Just to close that point, I think it's also an option available for them to reach out to other manufacturers as well. But since it's a private company, at this point, we don't know what the leaders at BennuBio are thinking, but they can definitely partner up with existing companies that have the ability to produce it at a larger scale. So maybe they can give permission for a limited amount of time, and with clear rules or like with clear expectations that we are planning to get like a feedback on how we can improve it and what else the market is expecting. That after that time period, whatever the revenues that we may make, we can increase on production, and also we can think about the second version of the product with the information that we can get from the customers through them.
Thank you, guys. We don't have time to get to the last two, but great presentation. And I just want to bring on Professor Marinakis and Linda and we want to congratulate you on tying for the first place team yesterday and getting that prize. And we are so happy to have you present to us.
Yay!
Thank you.
Thank you very much.
Hey, New Mexico sounds like a really interesting place. Do I need my passport to go there?
We'll have an exception. We'll write an exception for you.
All right. For me, the really interesting aspect of this technology is it's like the scanning tunneling microscope that gropes the surface to observe it. It's technoscience. It blurs the distinction between observing and intervening. It blurs the distinction between science and technology. And so it's wave of the future.
Literally.
Good job.
Thank you very much, guys, and congratulations. And judges, for this presentation, this was UNM Team 1.
And we'll see you tomorrow.
All righty. Thank you.
All right.
Good afternoon. This is UNM New Mexico Group 2. My name is Brian Fickett. The other members of this team is Clark Bird, Cory Carnes, and Taylor Uselman. Our mentor is Victor Chavez, and this is the case study for BennuBio. Next slide. BennuBio is a bioanalytical instrument firm based in Albuquerque, New Mexico. It's founded in 2018. The team has over 80 years of experience in flow cytometry research. Their co-product is Velocity. Velocity is an acoustically focused multi-stream flow cytometer. It counts cells in suspension based on given characteristics. It can process a large volume and has a high throughput, which will allow it to do big samples faster than the competition. The samples can be recovered after the process, which streamlines the current flow cytometry workflows. And it can count large cells and organoids, which is a plus and is not performed with other machines currently. Next slide. The challenges of scaling for BennuBio is on the short term is lack of scientific publications and demonstrating efficiencies of the velocity. Scientists are much more likely to purchase equipment that works well and will generate results. How best to show the scientific community that this velocity can revolutionize their research is the question. Long term, other firms may eventually add organoid and large particle functionality to the cytometers. This is could be an issue with BennuBio's long-term viability, and what else can BennuBio offer to customers to counteract this? I will turn this over to Taylor to go over the short-term goals, strategies.
Thank you, Brian. So, although the VeloCyte has tremendous applications in other fields, we think honing in on organoids and cell therapy markets provides the greatest potential. And our reasoning for this is that there's a vast array of subfields within these scientific areas, and both are really hot topics right now in biomedical research. So this is seen, for instance, in the global organoid market, which is expected to quadruple in revenue over up to 2 billion or over 2 billion by 2027. And it's also true for the US cell therapy market, where revenue is expected to reach 3 billion in the next 5 years. So collaborating with key opinion leaders and influential scientists on projects can help establish a sense of quality and meaning to your product and your firm, and that could help you penetrate these markets. So in doing research with these people, BennuBio can publish results on the VeloCyte's utility in a high-impact journal and reach hundreds of other researchers and future customers around the world. So this will also help develop expert user networks, which could provide scientific advisors, more customers, even pave the way for future funding and acquisition opportunities. We think the best way to do this is simply to reach out to them, loan them a VeloCyte, or ask for samples to get this validation research done. And this is especially true when BennuBio, or while BennuBio is in their current phase 2 funding stage with the NIH's SBIR grant. So on top of this, the Agency for Healthcare Research and Quality of the US, DHHS, can perform technology assessments for the VeloCyte. And this assessment can lead to your instrument being considered a gold standard for organoid flow cytometry research, and can provide a key advantage when competition grows. Next slide, please. So for our short-term scaling, again, like we said, we want to find you some highly influential people and groups, and that can cover the scientific fields of interest in various organizational settings. So for instance, in government, we recommend William Telford, who's a co-chair and of the National Institute of Health's Flow Cytometry Interest Group, and is a senior scientist at the National Cancer Institute. He also has many connections with other flow cytometry influencers. Additionally, Doug Melton and David Skadden at the Harvard Stem Cell Institute have tremendous access to organoid researchers at Harvard, as well as other key healthcare facilities and universities like MIT or Mass General, as well as other Mass General Hospital, I should say, as well as others around the world. And lastly, we highly recommend reaching out to Jeffrey Bluestone and other scientific leaders of the Parker Institute of Cancer Immunotherapy. Their whole mission encompasses developing novel tools and strategies to combat cancer by working with many of the top research facilities in the world. And the VeloCyte is one of those tools they could easily back. They currently partner with 2 other flow cytometry firms as well, but neither one of them have the capacity to analyze organoids or large particle patient samples like the VeloCyte can. So I'll turn it over to Clark to talk about the long term now.
Thank you, Taylor. So a primary challenge to scaling for BennuBio is that as scientists discover that flow cytometry for large particles and organoids is beneficial to their research, other companies will adapt and add this functionality to their machines eventually. So how can BennuBio innovate in the face of stiffer competition? As a biomedical scientist myself, I noticed that one feature that is typically synonymous with cell sorting was absent from the VeloCyte, which is a cell sorting function. So many flow cytometers not only analyze cells in a particle stream for a given trait, but actually sort them into different tubes based on this trait. This allows researchers to perform experiments on the purified cell populations. No one has ever been able to sort large populations of organoids before, and having this function could revolutionize organoid research as we know it. And now BennuBio does have a unique opportunity specific to New Mexico that we'd like to highlight to develop this sorting function. The New Mexico Small Business Assistance Program would pair with BennuBio with scientists and engineers from Los Alamos and Sandia National Laboratories to help develop this function at no cost to the company. So we think this would be an awesome opportunity to work within the state of New Mexico to improve the VeloCyte, especially working with Los Alamos, where flow cytometry was actually first developed. So why would sorting organoids be so revolutionary? The answer concerns advances in personalized medicine, which is a medical approach that takes into account an individual's genetic profile when tailoring treatments. This is important in the face of cancer, as each individual cancer is genetically unique, so tailoring treatment to specific mutations is vital to improving outcomes. Adding a sorting function to the VeloCyte will greatly aid drug discovery research. For example, scientists would culture organoids from hundreds of people with the same type of cancer, and then treat these organoids with a drug. They would then be run through the VeloCyte and sorted based on their responsiveness to this treatment. So once the organoids are sorted into their respective populations, genetic tests would be run to determine what genes mediate susceptibility to this particular drug. As you can see on the cartoon on the left, even though these two patients have the same type of cancer, they have two different mutations that are responsive to two different drugs. So identifying the drugs that are responsible for treating the different mutations is a critical aspect of cancer research. So what about funding BennuBio for the long term? We discovered that in the short term, they are very well funded right now with a combination of government grants and venture capital. But we thought that an interesting future funding mechanism would be to utilize crowdsourcing. So demonstrating that the VeloCyte is instrumental in drug discovery for cancer would generate a lot of positive publicity that can be translated into a public funding campaign. Appealing to the public's charitable nature is effective when getting money to fight cancer. So for example, the American Cancer Society typically raises over $700 million a year. So getting even a fraction of that money would be a great way for BennuBio to get additional capital. So BennuBio could choose to work with a crowdsourcing platform or establish a fund directly. But we found that one particularly interesting crowdfunding mechanism would be to take advantage of the recent rise in cryptocurrency by establishing it's called an initial coin offering, which is much like an IPO, just without all that regulatory hassle. So issuing your own cryptocurrency token would allow people to take a stake in your company and provide much needed funds for BennuBio's continued growth. So in summary, the challenge to growth for BennuBio in the short term is a lack of demonstrated efficacy in scientific citation using the VeloCyte, while a long-term challenge involves stiffer competition once competing cytometry companies adapt their machines to work with large particles. The short-term strategy is to connect with influential spheroid researchers, lending them your machines to get research done and published in top-tier journals. Others will adopt your technology once they see it can advance their own research. And the long-term scaling strategy to stay ahead of the competition is to add a sorting function to the VeloCyte, which would be a revolutionary new function that could greatly aid personalized medical applications. And lastly, an additional source of funding could be crowdsourcing, which would allow the public to contribute to your company's success. And so with that, thank you for listening to our presentation, and we'd be happy to entertain any questions.
Thank you so much, guys. That was a great presentation. I will just jump right into the questions. This is two questions in one, but I'm going to read both together. You suggested loaning equipment. How many are available or could be manufactured for this purpose? And how is this afforded by a small company?
Good question. Yeah, so I don't know exactly how many are currently available, but that is at a cost of $100,000 or selling at $100,000. The cost for developing the product to be a little bit cheaper than that, at least. So they would funding would need to be available to give out at least 3 or 4, whatever it would be, for what we mentioned for these research institutes. But the other way around is that instead of just lending them the machine and letting them use it, they could send us samples or send BennuBio samples to where you could do it on your 1 or 2 products and limit the amount that you have to actually send out to other places.
Yeah, a good alternative is sort of, instead of lending them as sort of a contract sort of research applications in which you would partner with these different researchers to get their samples and analyze them.
Thank you. How much investment do you expect would be required to add a sorting capability? And do you envision IP arising from this?
So that's a good question. So as far as the cost of developing that, it's not going to be a cheap function. I would say probably at least $1 million in development costs. Luckily, with that New Mexico Small Business Assistantship Program, a lot of those costs would be deferred using Sandia and Los Alamos laboratories. As far as IP, absolutely. Is sorting large particles, especially in the way that the VILA site does, would be a different application. So normally when you sort particles, it's kind of the droplets get charged positively and negatively to go into different tubes. Whereas the VILA site uses this acoustic harmonics to create this flow stream. So they might be utilizing a totally different technology in order to separate these guys into different tubes, which would obviously be a revolutionary new IP and be patent worthy. Heidi, are you muted? We see you muted on our screens. Oh.
She has stepped out, so I'm going to take it over.
Okay.
So sorry.
There she goes.
Are you suggesting the sorting concept back to Lannel?
As far as the development of it, I think it would be crucial to get it done with the scientists at Lannel, as they were the first to develop this flow cytometry back, I think it was the 70s. So they have a particular expertise in the subject. So working with them would be a critical way to go, I think.
And I think that like the team, BennuBio team does have like a lot of connections with that Lannel group anyways. So, I mean, if that's a possibility, that'd be great, working directly at Lannel or through other opportunities. Like I know various, working with the universities as well is often an option as well. So if there's opportunities for other people to get like engineers to help build experience or something, gain experience in internships or something to work towards a project like that too, it's like a double bonus in a way.
Thank you. NMSBA is limited to approximately $20,000 a year for companies in Albuquerque. There likely needs to be other funding to supplement. Have you considered?
Yeah. So I know I was looking at a couple of the investors in the Boston Analytics platform, and one of the ones the very first one actually that was the top investor in flow cytometry, to me, I thought was a very interesting option as well. In addition to these other, like we were mentioning, the crowdsourcing or the initial coin offering, but the Novo Holdings, they've been increasingly investing in more companies over these years, and as you can see, as they're increasing in the numbers of investments in total, they're also investing in quite a few flow cytometry companies. So I could see a company like that being, or Novo Holdings, for instance, being a good option as well.
Thank you. Cover again best candidates for partnering.
Yeah. Okay, so I mean, there are a ton of options, but I think that the Parker Institute for Cancer Immunotherapy is probably is a very, very good candidate, as there's the Parker Institute or the Parker Foundation itself provides a lot of money into cancer research that could open up doors for funding opportunities or whatever else may come from that. But for the research collaborations, some of the top researchers in the world of cancer therapy work with that institute. And organoids are like a novel upcoming scientific model to use for cancer therapies and diagnostics, and so that I think that's a great opportunity. as well as the Harvard Stem Cell Institute, because they are more focused specifically on organoids themselves.
Thank you. The application that you propose seems interesting. How long to the beginnings of product revenues?
So as far as once the sorting application is officially developed and proven to be useful, you're going to be able to get these in the hands of like big pharmaceutical companies who are doing drug discovery research really quickly, because doing these large scale drug discovery applications where you sort hundreds of different cells for subsequent genetic analyses is one of the primary ways in which people find what drugs and what genes mediate susceptibility to those drugs. So I envision that as soon as the sorting function has been demonstrated to be applicable and showing that and as researchers show that using organoids to perform this drug discovery research, it leads to better therapies, you're going to get the big pharmaceutical companies on board pretty quickly.
What is the status of FDA approval for this product, for the use of this product?
I am not particularly aware of FDA approval for this product right now, as it is a primary research mechanism. So as far as its use in drug discovery and then the FDA approval, I'm not sure.
Thank you. How about approaching pharma companies such as Lonus or Ionis?
Ionis Pharmaceuticals.
Right.
Yeah,
sorry about that.
I'm very familiar with Ionis Pharmaceuticals. I applied there once. So yeah, I think approaching them. So the Ionis is particularly interesting because they focus on mRNA sorts of mechanisms for treating disease. So if you could find specific genetic mechanisms that are mediating these cancers, Ionis Pharmaceuticals is actually a really good idea, because they are going to be the ones who are going to directly target those specific genes of interest using mRNA therapy. So whereas drugs can be kind of a dirty mechanism for fighting disease, these targeted mRNA immunotherapies are going to be cleaner and more efficient and better for the patients. So yeah, I would approach either like both the big pharmaceuticals and the smaller ones like Ionis who do work with mRNA.
Apparently the Ionis chief scientist is Bobo.
Oh, cool.
Yeah.
Have you considered the Broad Institute? Could be a research entry to both Harvard and MIT and a bridge to pharma.
Yeah, and it's a great idea. I think honestly, any major research institute like that, especially with top level universities, is a good idea. I think I don't know the exact research that they do in that particular institute, but I would say if it's related to the organoids or these cell therapies in cancer, that's a great idea, or even drug discovery.
And I would say that working with the big national institutes for health, especially the National Cancer Institute, is going to be a critical way to showing people that this really is a functional application for the VelaSite.
Thank you. We got about 45 seconds left for your last question. Sorting technology seems promising as a long-term strategic adaptation. Did you find outlets for the non-sorting technology as it currently exists for immediate revenue streams?
Absolutely. So the big thing about this machine is that flow cytometry, if I've done flow cytometry, it is painfully slow. So this machine works at a speed that is just out of control as far as these are concerned. So working with larger samples and finding better statistical representation of what you're actually finding in an experiment is huge, because sometimes you only find a couple of cells that are positive for what you want. Whereas if you have a large sample, you can get a better representation of what things are actually happening in the cellular process. So yeah, there's a lot of applications right now. And obviously this should be the primary marketing strategy as the machine stands right now.
Yeah. And even I would even say, even though you may not be able to sort a cell or sort an organoid, I don't know of any other large volume flow cytometer that can sort an organoid. There's none of the ones that we use in our core here at UNM. that use that. So to me, in that in itself is a really good application right away, just to characterize organoids via flow cytometry.
Great. Thank you, guys. You did awesome on your presentation. We are so glad that you are here, and we appreciate that. Thank you so much.
Thank you.
Thank you
Very much.
And judges, this is UNM Team 2. So our last student team is from Portland State University.
Okay, welcome to our presentation. It's about the BennuBio today. We can go another one. Yeah, I'm Aynur and our friends Angel, Ashwaq, and Ozgul. So our mentors are Professor Dr. Tuleyman and Dr. Kelly Cohen. So let's mention a little bit about BennuBio. It's a New Mexico-based startup. It's expanding the power of flow cytometry. And BennuBio can be a key game changer in the area of cellular analysis. The company has made some recent strategic changes, such as moving to a larger facility and selecting a new CEO, core product is Velocity, a new generation flow cytometer. So the Velocity, a new powerful analytical instrument that uses acoustic wave technology to analyze up to 10 streams. They speed 100 times faster than conventional flow cytometry, and no need for dilution of the analysis. And it can identify very rare cells like tumor cells. And it's a unique capability that analyzes micron to millimeter sized particles. So let me mention you some benefits of Velocide. This is a actual flow cytometric cost starts from the $30 to $100. The actual times goes up to 3 hours. The salary of the technicians is between the $67,000 to $71,500,000. If we would like to value perspective, the flow cytometry's value is $6.3 billion in 2020. The expected rate is $10.57 billion by 2026. Another some benefits of Velocide is can result with Velocide in 1.8 minutes, and the cost and time can be reduced with Velocide and used for further researchers. So if you look at the scaling strategy, the one of the important product line offering is the high throughput separate screening platform, and the pricing all these technologies is between the $100,000 and $150,000. The key potential funders in this area is the National Institute of General Medical Science and National Science Foundation. This is our business model canvas. We have prepared an initial business model canvas for the company. If the company is not currently using this tool, we recommend they do so and begin developing this in much more detail. Our graphic contains a lot of information and ideas, so we are going to emphasize just a few key points. The first, the continued focus on value proposition development is critically important for BennuBio. The company's scientists and managers need to further explain and connect improvements in equipment performance to customer value. To what extent do these performance increases translate into lower cost and higher value to customers? Currently, it is difficult to fully quantify and explain added value to existing customers, as well as understand how this product could open up opportunities for large additional markets in the future. Second, the company needs to carefully consider their existing revenue streams as well as several potential future revenue streams. It looked like direct sales of equipment has been one of their main current focus areas. We believe they should also consider leasing equipment as well as owning and operating their own equipment and charging fees to customers who want analytical test services performed without having to purchase or lease the equipment. This will allow them to take advantage of the machine's significant performance advantage to generate revenue and enable them to expand their manufacturing capabilities. It will also provide publicity for their product and build a base of customers who may one day purchase their equipment. Lastly, the BennuBio needs to carefully examine cost structure and elaborate on the other elements of the business model canvas that we have begun describing here. Those are our recommendations for this case, actually. So, can you move to the other slide, please? So, here's the, you can see the high throughput separate screening. This is one of the emerging technology in this area because BennuBio promises to usher in some possibilities in cellular analysis through its high throughput separate screening platform, which is on track for a 2022 launch. This is the new tool to study cell-to-cell interactions. This technology is a powerful multicellular separate 3D tissues tumor analysis system with potential application in 3D drug screening and other true high throughput screening applications. As I told you, this is one of the emerging areas, so that we did a scientometric analysis to identify the new emerging trends, the top authors, top institutions. So we came across, there are 10 patents in the Web of Science, and there are 11 patents in the Lancet.org. So another slide, please. So here you can see the research intensity with respect to the years. While the separates high throughput screening and the tissue engineering goes up in 2021, the others like drug screening, microfluidics goes down. So we can say there's need for fast screening and early detection of disease in the area. Another, please. So this is the emerging terms. While the between 2002 and 2017, high throughput screening and the separate was the top term in the area, these terms moved to the new, transformed to the new area like cholesterol cancer, invasion, metastasis, hypothesite, microval array, extracellular matrix, images analysis, personalized medicine. So focusing these new emerging terms, new emerging area, you can enlarge customer base and improve existing products. Even you can develop new products. You need more information about this area. So you can see here the top countries, top institutions, and top authors. USA is the best country in the area. So University of Michigan is the top institution in the area. So in terms of the authors, Looker, we can call him as a gatekeeper, is the best connection between the other connections. So another, please. So this is the main researchers and competitors. So Lebanon Chip has 33 papers in the area. And while the others like, I cannot see it well, sorry. While the John Edward and University of Michigan almost has 3 patents at the area. So you can establish partnership with research centers, institutes and faculty. Those are main researchers and the competitors in the area. Yeah, thank you.
Thank you very much. I am taking over moderating duties. Heidi's having a little technical difficulties. So at this point, we're going to ask our judges to starting asking questions in the Q&A section. Thank you very much for your wonderful presentation and for Portland State University in Oregon. Let's have some questions, please. First question. Do you have evidence that word count is correlated to growth?
Oh, sorry. Can you specify more the question? I mean, from the analysis that we presented at the end.
Okay, I'll go on to the next question. If Amy would restate the question. The various revenue streams you recommend. Explain why you believe software development is so important. Are you hearing me?
Yes.
Okay.
Yeah. So okay. Yeah. I think we don't have an answer for this question.
Okay. The scientometric analysis is a word count indicating the number of times a word is used, or am I understanding it incorrectly? Do you know this to be correlated with growth in that area?
Analysis is a word counting, so we use like a to understand what are the most, for example, cited the impact. We measure psychometric analysis.
I don't know that.
Yeah, I mean, like this can be correlated to the growth on the area. Let me see. If, for example, we have like a measure to a specific cluster. So depending of that measure, it's the value of that correlation. If the correlation is high, the level of centrality is going to be high. If there is not correlation, the level of centrality is going to be low.
Okay, we'll go to Steve Renfro's question next, since I missed that one. You described early funding from federal sources. Can you discuss your capture strategy for such funds?
Yeah, like for example, we recommended to work or get fund from the National Science, for example. So thinking that, for example, based on our analysis, that federal agency.
So that would be the first stage.
To expand the network of collaboration of, I mean, for our proposal.
Judges, let me know if you got all of that answer. You cut out a little bit, so I want to make sure that they got all of your answer. We have about 6 minutes left of questions. Angel, maybe you could answer that one again about early funding. Or Steve said he got the general idea, but maybe you could just say it again to make sure your capture strategy for your funds.
Yeah, like I was mentioned that, for example, we identify, for example, partnering with National Science Foundation. So that was one of our findings of our scientometric analysis. So the other good finding is what my teammate Aynur explained, that, for example, the University of Michigan was one of the main universities that is working in this specific topic. So I think that the analysis show like what's the best route to start making like a network of connection, working with the academia, working with companies, or working with government organizations.
Thank you. The cost analysis was compelling. So what's the strategy to present those cost-saving possibilities to commercial laboratory operators?
Yeah, we did a little bit of research. What is the current situation? And identify that from the not sure, like how in terms of money, how beneficial this could customers. So that's why we are in our campus model. Like we identify exact proposition for new customers. So that's we include that part in our campus model. I don't know if
on how we're having a great deal of trouble understanding you. Can one of your other team members answer the question?
So let me answer the question about the scientometrics. So we use, of course, the word count for identifying the trends. We do not have evidence that it's correlated to growth. When we are using the scientometric analysis, we are identifying some keywords and then searching on the Web of Science or some patent web pages. And then depending on our results, we are doing some analysis like collaboration network, Contreras network, something like that. And then the results actually the trend topics, trend technologies, and something like that.
Angel, was suggested by your professor, maybe if you remove your headset, that is where the interference is coming from.
Oh, can you hear me?
Yes.
Okay.
So maybe you could say again, answer Ms. Donaldson's question about the strategy to present the cost-saving possibilities to commercial laboratories.
Oh, okay. Yeah. So I was having to get the question. We like a new technology. That's all I can say. Like about the salary of the use these resources. Develop like a new product or use the resources to put the money in other areas that can help, for example, need to grow
It's good money.
We have about 1 minute left. Do you know the status of FDA approval?
No, at this
Okay, thank you guys. That's the end of our time. We appreciate your presentation and your hard work, and you guys did a great job. Thank you. Judges, again, this is Portland State University.
Great
Thank you so
job,
much.
Portland State. Thank you for joining us.
thank you for your time.